there, we're going to take a look at activity 4.1 today. So we have some experimental tools here and we have our handy dandy friend. Hi, I'm Liz. And we're going to go ahead and check this first bit. We're going to be using indicators today. The question we're trying to investigate is what's happening when starch gets broken down. So you may have heard of a food being starchy, right? Crackers are a great example. Pasta. You may have heard of potatoes being starchy. So we're going to take a look at what happens when starch breaks down. In order to do that, we need to figure out how to use these chemicals to see what they test for. So first, we're going to take a look at something called Benedict's solution. Now, Benedict's solution is very pretty. It looks sort of like mouthwash. Don't ever drink it. It's pretty hazardous. We're going to put some Benedict's solution into some vials we've prepared ahead of time. We have these little microtubules, and one of them is for water, one for glucose, and one for starch. So for the first data table, it says record the color Benedict's solution, a test for simple sugar turns when it goes with each of the solutions. I'm going to put 5 drops of Benedict's solution in. Now, one thing that's particular about Benedict's solution, Lizzie, is that this hasn't reacted yet. What do I need to do to make Benedict's react? I don't know. I'm going to heat it up. So we have this big beaker of water sitting here getting quite warm, and we're going to put our microtubules that have Benedict's solution into this beaker, and we'll let it warm for 5 to 10 minutes. We don't want to boil them. We just want to heat them. So when we start off with these, with the Benedict's solution in them, they are blue, a very light blue, like we see in the Benedict's solution. It looks just like it's been diluted. We're going to allow these to heat for 5 to 10 minutes. While we're doing that, we're going to check the effects of iodine. So Lizzie, if you wouldn't mind, we have here water, glucose, and starch, already measured out, and we're going to put about 1 drop of iodine into each tubule, and we'll take a look to see what happens to the color. Right now, they all started off clear.
So the water, it's a very pale yellow, just looks like the iodine's been diluted. With the glucose, it looks yellow, also like it's been diluted. Ooh, when you put it in the starch, it turns this very vivid brown color.
It's almost like a purpley brown.
Yeah.
Okay, can I have those over here to take a picture? All right, so through the magic of the internet, we've waited about 5 minutes here, a little more, and we can see a change in these tubes. So I'm going to go ahead and fish them out. And these are the tubes we had with the Benedict's solution in them. And our book tells us that Benedict's solution tests for simple sugars. So if we take a look at these, Lizzie, if you could describe them, I'll write what you say. So for the water,
it's still a very light blue, like the Benedict's solution.
okay. For the glucose,
It's turned almost a brown, sort of yellow color.
okay.
And the starch, it's also blue, like the Benedict's.
So the only one that really shows us a change would be glucose. So glucose is a very simple sugar, and the way Benedict shows us that it has found the glucose is that it turns this yellow color. Okay, great. So now we're going to take a larger look. Now that we know how the tests come out, we want to see what happens when we have a variety of things. So first we're going to test for glucose with Benedict's solution. And to test for glucose with Benedict's solution, Lizzie, you prepared some tubules for us.
Yep.
Tell us about them.
So the first one has 10 drops of water and 5 drops of Benedict's solution.
Hang on a second. We know what happens with Benedict's solution in water. Why are we testing this again?
It's a control.
Tell us about a control.
Well, a control is to make sure that nothing from the test itself is directly impacting it, other than what we want to test for.
We're looking to make sure what we're testing for is really what we're testing for, that we're doing the experiment correctly.
Yeah, that there's no outside factors playing into this.
Wow, that's so grad school. Okay. All right, so we're going to go ahead and put our microtubule with water. And we don't need to observe color before in our data table, but it's light blue.
Next. The next one is 10 drops of starch solution and 5 drops of Benedict solution.
Okay, now we saw before, again, starch is not going to change color. Why are we doing this?
It's another control.
It gives us something to compare to.
Mm-hmm.
Okay, great. And then
we've got 10 drops of glucose solution and 5 drops of Benedict solution.
Okay, and we've done this before too, right? We know that glucose is the only thing that's going to change color when Benedict reacts. All right, now
we've got 10 drops of cracker solution.
So let's talk about how you did this.
Sure thing.
You took some cracker,
yeah,
and then what?
And I ground it up in a mortar and pestle, and then I added water to it and took 10 drops of that solution.
Okay, so we're just testing cracker and water here.
Mm-hmm.
All right, go ahead and put that in. And then last. So taking a look at our last two, what's in there?
So this one also has 10 drops of cracker solution. It has 5 drops of Benedict solution, and I'm going to add 10 drops of our enzyme alpha amylase.
So amylase is an enzyme. Now, enzymes can be found in our bodies. You may have heard about this before. Specifically, one place we find them is in our saliva or in our mouth. So this amylase enzyme did not come from our mouths, came from a jar, but we're going to use it to simulate what would happen if you chewed up the cracker. So just to be clear, in tube 4, you have water and cracker. In this case, you have water, cracker, and an enzyme.
Mm-hmm.
An enzyme is a catalyst. We've talked about catalysts before.
Mm-hmm.
Catalysts don't take place in the reaction, but they help the reaction happen.
Mm-hmm.
So when we're looking to see like magnesium oxide, we burned that strip at school and it turned bright white. When we did that, we said the flame, that match, was part of a catalyst. All right, we're going to allow these guys to heat for about 5 to 10 minutes and then take a look at our results. While we're doing that, let's move on to the iodine, and I think you have those set up as well.
Yep. So, again, in my first one, I have just water.
And this serves as a what? Control. Because scientists may be lazy, but we don't want to do this experiment twice. We want to make sure what we're testing is what we think we're testing. Now, does iodine need heat, Lizzie? No, we don't need to heat this one. So we have results right away. Tell us about what happens when we have that iodine and water.
So again, like we saw with the samples, the iodine just looks diluted. It's still a very pale yellow.
Okay, and I'm going to make this adjustment. It may say in your book color after warm water bath, but we said we don't need to put iodine in warm water bath, so we're just saying color after. And you said that was kind of a yellow.
Mm-hmm.
Okay. Is starch present?
No, not in the first one.
How do we know that?
Because if starch was present, it would turn that purpley brown.
Okay, so we're going to go ahead and put no. Okay, and for 2B?
So for 2B, this one is starch solution.
Okay, so hopefully we know what should happen there. If our test is working correctly, iodine will detect the starch. And
it absolutely did. It's a purpley brown again.
Okay. And so starch was present.
Mm-hmm.
All right, for our third one,
we have glucose.
Now, glucose is a simple sugar. Glucose can build up into starches, or starches can break down into glucose. But should we detect any starch here? Probably not.
And no starch was detected. It's, again, a pale yellow.
Okay. And then
the fourth one, like our Benedict solution, has just the water and the cracker.
Okay.
already see up at the top that it's become purpley brown. So there's definitely starch detected.
That's cool. I'm going to take a picture.
Now this last one is going to have the cracker and the water, but we're also going to add our alpha amylase enzyme again.
All right, so adding that amylase enzyme, again, simulates what would happen if you chewed up the cracker and spit into the tube. We don't want to be so unhygienic, and we don't want to contribute our DNA to the lab, so we're going to use this manufactured amylase, but it does the same thing as the enzyme in your mouth.
Shake. Oh my goodness, and then almost exactly, it turns that purpley brown again.
Okay, so we see some starch. If we give that some time, do you think we're going to continue to see it? I'm not sure. All right, so let's take a look now at our results from our test with the Benedict's solution. All right, I'll pull these tubules out, and I'm going to ask you to write down the data this time, Liz.
Sure thing.
Okay, so tube 1A, which was just water, there's no change. It's blue. Our control worked. Since it's blue, that means no sugar was detected. Okay, for tube 2, which was our starch, correct? Okay, this is also blue. Again, no sugar detected. For 3, glucose. Glucose is a simple sugar. We should be finding it. And sure enough, Benedict's turns this bright orange color.
Ooh.
Definitely found some sugars there. For tube 4, we have our cracker and water, and it doesn't detect any sugar whatsoever. Finally, for tube 5, we have our cracker, and this is with the special enzyme in there to help start breaking down the cracker. And we can see when you break down the cracker, it turns this yellow color. So we're definitely seeing that the sugar is present. All right, so that's our data for today. We'll post a picture of that for you online, or in this video even. Once you've done that work, there are some making sense questions for you to complete. So thanks very much, everyone.
Bye guys.
Bye.